Hello, my name is Catherine Castrillon. I'm a graduate student research assistant at South Florida Terrestrial Ecosystems Lab at Florida International University, and this is a tutorial on gap light analyzer for hemispherical photos. So gap light analyzer is an imaging software used to extract forest canopy structure and gap light transmission indices from true color hemispherical photographs utilizing fisheye lens. It computes canopy, site openness, and leaf area index, as well as other factors. Gap light analyzer, which I'll be abbreviating to GLA, includes a number of tools and image processing functions to register and manipulate the imported images. These image processing functions are threshold, brightness, contrast, which can be used to affect the entire image or only a select portion of it. An important factor to consider with utilizing GLA is organization of your workspace. At Softel, we take hemispherical photos for tree islands in the Everglades National Park and the mesocosm tree islands at Loxahatchee Impoundment Landscape Assessment, otherwise known as LILA. For instance, this image shows the directory to locate hemispherical photos for tree islands within the Everglades National Park. Each folder here contains hemispherical photos for every field season that those tree islands were sampled in. In this tutorial, we will be looking at Grossman Hammock located in the Everglades National Park. Grossman Hammock contains photos for field seasons ranging from 2010 to 2020. If photos for a field season have been processed, they should contain an original photos folder and a processed photos folder. These photos should contain the following. The original photos folder contains all hemispherical photos taken within the same field day for the specified tree island. The datasheet contains raw information taken in the field. Here the datasheet shows that 33 photos were taken in total. Tree island's cell number and photo ID given by the camera is noted. So the original photos folder should contain 33 photos as well, and it does. Before moving on to the processed photos folder, which contains information given by gap light analyzer for images selected, I want to review the image selection process. As discussed, we have 33 photos. However, we have 16 cells total for Grossman. Only one photo can be selected per cell, and that photo will undergo image processing in GLA. The total output for Grossman is 16 processed images and 16 summaries, which I'll get into momentarily. So here we have our list of images from our field day datasheets. A minimum of 2 photos were taken for each cell. For image processing, only 1 photo per cell is selected, and the photo selected should have little to no sunlight interference, glare, reflection of light on leaves, branches, and main stems, which at times may not be picked up by GLA image threshold, among other things. We'll go more into this once we dive into the program itself. For cell 1, let's take a look at 2 photos which were taken, 5066 and 5067. If we take a look at both images side by side, which image would be best to work with in GLA? If you guessed image 5066, you're correct, as image 5067 has sunlight interference. Both images have reflected light on the main stems of the trees, but this can be fixed. Sometimes both photos may have this same issue. In that case, we have to fix the main stem to the best of our ability. And I'll review that once we open up the gap light analyzer software. To track all images chosen per cell and progress made on them, an Excel sheet will be created per tree island and field season. Here we have a blank data sheet to track our progress. At the very top, we will enter the tree island we have been working on, which is Grossman Tree Island for field season 2017 to 2018. The photo ID pertains to the number of photos taken that day. We took 33 photos. So this is 1 and 2 for cell 1, 3 and 4 for cell 2, 5 and 6 for cell 3, and so on, as you can see by the data sheet. Photo chosen pertains to which photo will be processed. For cell 1, as we had spoken, we chose photo 5066, and that was the first photo. Photo original JPEG name is DSCN, which is camera code, and 5066, which is the image that we chose. Eventually, after processing, this photo will be named GH for Grossman Hammock _ cell 01. And we also enter the day that the photo was taken, which is January 31st, 2018. Eventually, we enter the date that the photo was processed in GAP Analyzer. Here I have a completed sheet, and you can see here that it took 2 days to process these photos in GAP Analyzer. Sometimes it can take a little bit more than 2 days or even 3, and that's okay. So to review, the original photos folder contains all hemispherical photos taken within the same field day for the specified tree island. Usually about 2 to 3 photos are taken per cell of the tree island plot. Number of cells are dependent on the tree island. Grossman has 16 cells, so only 1 photo should be selected per cell for processing through GLA. Photos selected to be processed are placed in the same area as these 2 other folders, as you can see here. And the progress made on the processed photos will be noted using Microsoft Excel, seen at the very bottom. The processed photos folder contains 3 folders: configuration, summary, and working. The configuration folder will contain the configuration created prior to working on an image in GAP Analyzer. The configuration is dependent on the date photos were taken, location, such as latitude and longitude, magnetic correction, as well as other environmental factors, which will make more sense when we enter GLA. Usually there is only 1 configuration created, as all photos should be taken on the same day. If photos were taken on different days for some reason, then a new configuration will need to be created for those photos only. The summary folder contains the calculations for canopy structure and transmitted GAP light log details for each process photo completed per cell. The summary files contain % canopy openness, % site openness, leaf area index, and more. And the working folder contains the finished process image, which will look like the following. We began with image 5066 on the left, and after using image processing functions from GAP Analyzer, such as threshold, brightness, contrast, and so on, our output is GH _ cell 01. With that, let's jump into GAP Analyzer software and go through the protocol for analyzing hemispherical photos step by step. And remember that organization is a very important factor here.